Hi, my name is Grace. I will be presenting my journal club article on the impact of PPIs on GERD patients' microbiomes. This research was conducted in 2019, and the name of the article is The Effects of Proton Pump Inhibitors on the Gastrointestinal Microbiota in Gastroesophageal Reflux Disease. So, for the bluff, the main research question is: does the use of proton pump inhibitors impact the gastrointestinal microbiota of GERD patients? So, researchers went about answering this question by collecting samples from a variety of patients using gastric mucosal and fecal samples, which are from the stomach and the intestines, sequenced the bacteria found with Illumina, and identified them using the 16S rRNA gene, which we learned this week is very important due to the highly conserved and highly variable regions. And the region they selected is the B4 region. A little bit more background, just so this all makes sense. GERD is a condition in which stomach acid repeatedly flows back up into the esophagus. So, as you can imagine, that's extremely uncomfortable. And people take proton pump inhibitors to reduce the volume and increase the stomach pH of this acid. And basically, it works by literally inhibiting the proton pumps that are pumping all these hydrogen ions into your stomach. So, this research is urgent because it could be really negatively impacting patients long term, and it's not something that I think a lot of people realize. And maybe this research will show patients that other options may be well suited. So, to dive a little bit deeper into the background, I want to go into the past work. And one of the earliest studies I could find that addressed this issue was from 2009. And it said that PPIs impact the flora of the microbiome by raising pH, which we already knew this. This was becoming more basic, as we learned in the last slide. But it specifically hurts the bacteria that might be acid producing in gastroesophageal regions. So we get a lot of help with digestion from bacteria in these regions. And if the optimal pH for this bacteria is not being met, let's say it's more basic, like a PPI would do, it could either die or become less fruitful. Another study I thought was very interesting showed how PPIs are being inappropriately used. So basically, they were designed only to be taken short term, but more recently, people have been taking them long term, which has been causing a bunch of issues. And when they stop, they return to their original GERD symptoms. So even though we knew all this information, this study addresses kind of what bacteria are being eradicated by the PPI and their implications. In order for researchers to determine the impact of PPI on the gut microbiome, what bacteria were being killed off, and the degree of change between short-term and long-term PPI users with the fecal and gastric mucosal samples, they first collected the samples and had them sequenced by Illumina, identified them by their 16S rRNA gene, and they ran lots of statistical tests on the data to determine the alpha and beta diversity of the gut and gastric mucosal samples. So, something I didn't know before reading this article was the difference between alpha diversity and beta diversity. So, alpha diversity is the diversity within a sample or within a community, and beta is between samples. So after that, they procured a list of the most common bacteria present in each sample, and between PPI users, non-PPI users, and healthy controls, they also identified what was most prominent in each sample and the volume that was usually in that area between the stomach and the intestine. And then they discussed the implications of having increased or decreased levels of certain bacteria and diversity of bacteria in PPI users compared with healthy controls. Some strengths that the researchers used in this area were choosing Illumina as their sequencing platform. So Illumina has a low price per base, provides high quality bases, and has a massive throughput, which was perfect for the study of sequencing many different kinds of bacteria. And researchers use set time frames and standards for groups. So long term and short term are kind of vague expressions, and they chose very specific time periods for individuals being sorted into those groups. Some limitations that I noticed were that the participant pool was kind of small. There was only 35 participants for the gastric mucosal samples and 55 for fecal, and everyone was between 46 and 54. I think I would have appreciated if there was a wider age range And they noted that there was possibility of contamination between the gastric and fecal samples because they are so close together. To jump right into the results, the researchers first addressed the gastric mucosal alpha and beta diversities. So, to begin with the alpha diversity of each sample, the first statistical test that they used was the KO1 test, which measures species richness, and the GERD minus PPI condition showed a higher KO1 score than GERD plus PPI condition participants, which means that it predicted more unseen species and a higher species richness than the GERD plus PPI condition. Um, Next is the Simpson Diversity Index, and this showed that the healthy controls have the highest amount because, first of all, there's not many outliers. The mean is right in above all the other ones listed there, and the values are really controlled in that tiny little box and whiskers plot. And if you see, the GERD minus PPI condition is next, with the mean being slightly below, and finally the GERD plus PPI condition. So this is aligns with what we saw in the KO1 test, and that even the GERD plus PPI condition has many outliers, even below zero point 0.9, that are kind of alarming and different from the GERD minus PPI. Obviously, you like to see that the healthy control scores are all localized up near 1. Now, an interesting result that I thought was significant was that when they performed the Shannon index, which is another statistical test, the p-score was over 0.05. So they kind of decided that the similar overall microbial diversity between the groups was maybe because that test also addresses species evenness. So even though the species and like amount of bacteria may be even across the area, the diversity is different. And finally, we have our beta diversity here to the right, which is the PCOA1 test. And that shows that GERD patients and healthy controls have distinct gastric mucosal microbiomes. So as you can see, the boxes are completely separate from each other, which again, exactly what we'd expect in our results. That the healthy control was way out here in front of everyone and had the highest score pretty much localized to that small box with hardly any outliers. Moving on to the bacteria that they found in the gut, to first address the alpha diversity seen here, the KO1 and the diversity index showed that the healthy control is the highest and has the highest level of diversity. This aligns with what we saw in the last slide. The only difference is that the KO1 test here addresses the healthy control, and the KO1 test on the last slide that I had just addressed in the text only included the GERD minus PPI and the GERD plus PPI conditions. So, of course, the GERD minus PPI condition had a higher rate of diversity, but here, when put up against the healthy control, you can see that the healthy control has way more diversity, which we already knew that just regular GERD patients already have a less diverse microbiome than healthy controls. And even though I was addressing the KO1 index, you can see that the results are almost exactly mirrored in the diversity index, and that even the outliers for the healthy control are still most of the time above any other condition. Now, moving on to the beta diversity, this is what I thought was specifically very interesting about this study, and it'll be addressed later on too, that the GERD minus PPI and GERD plus PPI samples showed no statistically significant differences, which is seen in this PCOA2 graph right here. You can see that the boxes are looking very similar, the orange and the green, and they predicted that this was because the PPI works directly in the stomach. So obviously the vast differences in microbiota are going to be more well seen in the stomach than in the gut. But still, the healthy control has the highest level of diversity here too. You can see that the mean is above the rest of them. So, as for the microbial composition characteristics, they took the samples, sequenced them, and basically in these charts, they showed the main phyla in each condition. And even though they look very similar, for the top graph anyway. This is just the phyla, so there's a lot of differentiation between the genus and species, which you'll actually see in a cladogram on the next page. But it was still kind of interesting to note that while they appear similar, the different genus and species of each bacteria actually causes a significant difference. Also interesting to me was that the fecal microbiota seemed to have a much different makeup than the gastric one. That's not really something I guess I would have thought of, but the two different areas play different roles in the body. But as you can see, even for the GERD plus PPI condition, the different amount of bacteria is still lower for each condition, for the fecal and the gastric samples. And down here at the bottom, I know it's really small, but it's actually very cool to see the abundance of the 35 most dominant bacteria in each condition. So really small at the bottom, that you can see that the first bunch is the GERD minus PPI, the next is GERD plus PPI, and then the next is healthy control. And my main takeaways from this graph were, first of all, like, wow, this is such a fascinating look into our microbiome, but also that the healthy control has the most uniform set of bars. So if you look closely at all the way to the right-hand side, even though they vary, they look mostly uniform compared to the GERD plus PPI condition that's just all over the place, directly to the left. You can see all the different colors and markings, and that's just all different samples from different participants, but it definitely does not help to have such all over the place results like that. And here's that cladogram that I mentioned on the last slide. So if we think back to the last slide, I had noted that the phyla were addressed, and that's why the bar graphs look very similar on their composition. To me, I thought they looked very similar, even though there are some distinct differences. But when I saw this cladogram, it kind of made more sense to me about the different diversity. The more branching that one of these conditions has shows that it's more diverse and shows really, truly all the tiny different little details of the different bacteria going in. So, as we think, the healthy control obviously has the widest band and the most branching involved. That shows the most broad diversity, not just in the minor little different bacterias, but just overall, even community-wise. This is followed by the small green bands, but you still see that, especially down at the bottom, it shows lots of branching in the bottom and the bottom right, just a little bit. Finally, the last condition is the GERD plus PPI, which you can see is very little branching. This is just evident in everything that we've seen before of the decreased diversity. And both this cladogram is for both the gastric and intestinal regions. So this just further corroborates everything that the researchers were trying to figure out. And if you look at the figure below, I thought this was probably one of the most interesting figures in the article that I had read, because it correlated all the little bacteria you see in the right of the first figure, all the different kinds of bacteria and what's most prevalent in each section, and related it to what the basically main roles were in the healthy controls, the GERD plus PPI and GERD minus PPI. So you see for the healthy controls, the most prevalent activity going on is the polycyclic aromatic hydrocarbon degradation. And this basically just means that healthy controls are super good, and the bacteria in them are super good at breaking down toxins that the body has, which can protect against everything as diverse as DNA damage and risk of cancers to inflammation. And next, in the GERD minus PPI condition, what stood out to me was the methane metabolism, which is obviously good because we know a lot of methane goes through our intestines, but also the terpenoid backbone biosynthesis I saw led to the synthesis of things like bile acids. And so I believe that this would cause more acidity even in that region, which would make sense for a GERD patient. And finally, the GERD plus PPI condition has a high level of glutathione metabolism, which could indicate oxidative stress. So, as we would guess, this would be a very bad thing for our intestines and would cause not as good digestion as seen in the healthy controls or even GERD minus PPI conditions. So, finally, moving away from the diversity differences between the different conditions, like the healthy control and stuff, we are moving on to what I found to be the most fascinating, the bacterial differences in short term versus long term PPI usage. So, kind of the trend that I noticed and that researchers had also noted was that the short term PPI users would have less helpful bacteria, and the long term PPI users would actually have a buildup of potentially not so much harmful, but an overabundance kind of harmful bacteria that is seen right here in the results. So basically, short term PPI users had less bacteria like the Pelobacter, Desulfuromonas, and Alcanivivorax than healthy control. Um, and the long term PPI users had higher levels of Luteomonas, Limonobacter, and Herbaspirillum. One that I thought was particularly fascinating was the Methylophilus that was found more in long term PPI users than short term, which the other ones were harmful and overabundance, but this one particularly I thought would be more helpful because it assists in breaking down methylated compounds and contributes to like nitrogen cycling in the gut. So while some of them were bad, this one happened to be good. I don't know if that was maybe just in this study or if that's in most participants, but I thought that was really interesting. As for the gut, um, healthy controls have a larger amount of diverse bacteria communities, and we already know that. We saw that in the last several result slides. But they also have a specific one, the Ruminococcus. And Ruminococcus is actually helpful in maintaining intestinal health by digesting fibers and such. So we see down at the bottom that the GERD minus PPI condition has way more Ruminococcus than even the GERD plus short term PPI condition. So obviously, healthy controls were the highest, and then it goes down the more from GERD to users of PPI. Also, I thought it was interesting that in the gut, it was a little bit different than what we saw in the gastric mucosal samples, was that the short-term users had more streptococcus, which you see in the graph down below, than the GERD minus PPI, which is actually harmful in overabundance. And then, just like we saw on the other side, long-term users had an overabundance of Veliono, Lella, Kake, Micro, Caca, and associated bacteria, which, sorry, they're just really hard to say, but this negatively impacts digestion for these people because these bacteria are directly competing for resources that healthy bacteria would need to survive and strengthen the intestinal health. I believe the authors did successfully determine that there are microbial changes in PPI users, non PPI users, and healthy controls, along with PPI users for short term and long term amounts of time. One of the main results that we have here is that long term PPI use causes more drastic changes in the microbiome than short term use. It increases certain bacteria like Streptococcus, but decreases some like Pilobacter. So basically, this is saying that the longer amount of time that the pH is changed and the volume of stomach acid has changed, the more detrimental the deficits. Also, that PPI use decreases microbiota diversity in the stomach, but not as much the gut. This is because the most of the proton pumps in your body are in the stomach, while there are some throughout your digestive system. Most of them are in the stomach. So obviously the gastric mucosal samples will be the most impacted, and that was seen in the results that we went over a few slides ago, that some of the gut samples weren't even statistically significantly different than each other. And finally, healthy controls have more diversity than individuals with GERD. I believe this data that we obtained, even though we already knew this, I think that identifying the bacteria that lack between the two could be a step in the right direction of medication that does not involve PPI, that but also improves GERD, maybe even like a probiotic. And I think it's important to note the strengths and weaknesses of not only the methodology, but also the results. So, the biggest strength of this was that they obtained evidence to support their research question and their hypothesis that even though the Shannon index came back as not statistically significant, in the alpha diversity measure of I think, the gastric mucosal microbiota. All the rest did. And I really think that lends to the credibility of this research article. But one of the major limitations that even the article noted was that they did not find PPI use correlated to a higher risk of gut related infections like C. diff, which is fascinating because that Taewon study I listed underneath shows that out of the 54 most recent studies done in this area, most of them, like a very high number of them, relate the gut related infections to PPI use. So I think that more reflection needs to be done with the data to see if that is more of a possibility that they overlooked. And also, they noted that future research would be needed for the H. pylori pathogenesis of GERD. They did not go into depth with this subject. They noted H. pylori and how it can cause gut related issues, but not as much into the pathogenesis of GERD. So I think future studies should take that into account and maybe use this data to put towards that subject. To conclude, I think that these results are very significant to today's medical field, especially for GI doctors, so they can let their patients know about the potential impacts of PPIs on their gastric and fecal microbiota before they decide to take it. Patient education is so important because everyone has a right to know what a medication will do inside their body. So I appreciate that doctors and researchers can now tell their patients that PPI significantly change the species richness and diversity in the gut and intestines, which are both crucial for digestion. I believe these results will also be big in the pharmaceutical community because I think that there should be a call for a less harmful type of medication for GERD patients that will still provide them with relief, but without damaging their microbiome. Here are some questions that I thought up for you guys to answer about the study and the research that we just went over. And here are the articles that I referenced in my journal club presentation. Thank you guys so much for watching, and I hope that this makes you think a little bit deeper about your gut microbiome and what makes it up. Thank you.